Let's see now the small intestine. And for the small intestine, it has the three parts: the duodenum, the jejunum, and the ileum. Now, all of them are very similar in terms of structure and in terms of function. Okay, the names are coming from the Latin time. When they first dissected a human being, they noticed, or maybe the cat, they noticed like three different colors, and that's why they called them three different names. But in general, all of them are very similar in structure, and all of them perform the same function, which is the big function of the small intestine is the chemical digestion and, importantly, absorption. So most of the absorption of the nutrients are happening in the small intestine. Now, to allow this small intestine to do those two processes, there are modifications happen in the small intestine, and technically there are three modifications. The first one is called the circular folds. And what is it? It's the bending or the folding of the mucosa layer in the small intestine, the foldings in the mucosa layer. And the circular folds will serve two purposes. First one is slow down the chyme and therefore allow time for chemical digestion and absorption. And the second reason, or the second function, or the second purpose is to increase surface contact with chyme. So we will have better efficiency for the chemical digestion and absorption. What do we mean by increasing the surface contact? I need you to think of those two areas. Let's assume that this is the mucosa and it's flat and the chyme is here. So those cells here will have just their surface to come in contact with chyme and start to absorb nutrients versus this mucosa. Okay, so we have here the cells. So we'll have more cells along those folds, and therefore we will have more surface contact with the chyme. Okay, that is what do we mean by increasing the surface contact. And this was for the circular folds. Then the second modification, that within those circular folds, we have additional folds. So here, the entire mucosa is folding. But now, even the very superficial layer of the mucosa, also it has its own folds. And those folds will form like finger-like structures. Those will call them the villi. The villi. Each one of those is a villi. And also those to increase the surface contact. So remember here, again, that we have those cells. So instead of just laying those cells here, now I will create another folds, and those are the villi. So think of them like a double fold. Okay, now then the third modification is the microvilli. The microvilli. What do I mean by the microvilli? If I draw or erase this, so the circular fold, then inside we'll have the villi. So I'm going to draw one villi. So this is one villi. On the surface of the villi, we have those cells. Okay? Those cells on their surface, they will have the microvilli. So those are cellular extensions from the cells, columnar cells. Those are columnar cells. So in their surface, they have the microvilli, or sometimes we call them the brush border. What do they do? Also increase surface contact. So three modifications in the small intestine to better perform its two functions, the chemical digestion and absorption. First one, circular folds, slow down the chyme and increase surface contact. The villi, the folding of the superficial layer of the mucosa. Then the microvilli on the surface of those cells. So those are columnar cells, simple columnar cells, epithelial cells for absorption. Okay, so the absorption of the nutrients will happen through those microvilli. What else do we have there? We have another type of cells called the goblet cells. Those goblet cells are exocrine glands. So each cell is a single exocrine gland. What do they produce? They produce a mucus. And the mucus here is important because it's going to wrap up the chyme. It will make its surface smooth so it runs over the mucosa. Additionally, in the intestine, we have something called intestinal crypt, which is kind of inverted villi. This is the villi. This is think of it inverted villi. Usually what do we have here? We have stem cells. Those stem cells would produce and regenerate the columnar layers or the columnar cells, because those cells usually consumed, eaten up because of different chemicals coming through the small intestine. Some of those are enzymes that will break down proteins coming from the stomach and also coming from those same cells. So those cells are constantly being consumed, so we need always to replace them, and this replacement coming from those intestinal crypt. So they are inside the crypt, kind of protecting them from the chyme. Inside the intestinal crypt, we have another group of cells called enteroendocrine cells. And those are responsible for producing digestive enzymes. So digestive enzymes will go up into the chyme, get activated here, and then, start to help in the chemical digestion. Those cells are also here coming from the intestinal crypt. In the small intestine, there is a small portion of the digestion is mechanical because of the movement of the circular and longitudinal muscles. So there will be like segmentation. So when muscles contract and then relaxes, okay, they alternate in their contraction and relaxation. And therefore, they will segment those pulps and remix them again. And this will enhance the chemical digestion. Then, all of this, the remaining of the chyme, would move to the large intestine. Large intestine, its biggest function is to reabsorb water and therefore solidify wastes, producing feces. So those chyme, or the remaining of the chyme, will go to the small intestine through a valve. There is a valve here called ileocecal valve. Ileocecal valve. This is between small intestine, or the ileum of the small intestine, and the large intestine. Okay? It's here, ileocecal valve. So the chyme, once it's in the passing this valve, it will reach to the cecum. Cecum is think of it like a small bag, a small sack, that most of water is being reabsorbed there. So the first process of solidifying waste is going to happen in the cecum. Of the cecum, we have the appendix. And the appendix, initially, we thought that there is no clear function for the appendix, but later it turned out that it hosts microbial flora, which is those microbes of the good bacteria in our large intestine that will help in digestion as well, producing enzyme and producing vitamins, mainly vitamin synthesis. So those will eat some of the remaining undigested food that we consume and turn it into something useful. Okay, so that is for the chyme. Once it is there, it will process through the transverse colon, through the ascending colon, then the transverse, then the descending, all the way to the sigmoid, and then to the rectum. The rectum will serve as temporary storage of feces. Okay, when we are ready to defecate, it will go to the anal canal or through the anal canal and then outside the body. But the point is, as this chyme is moving forward, it will get solidified and we will kind of concentrate those wastes. So in the large intestine, we don't really have a villi as we saw it in the small intestine. There are some cells with microvilli, but mainly what we will see is goblet cells. All of those gray dots or gray circles, all of those are goblet cells, and we will see them under microscope. So here we have plenty of goblet cells to produce plenty of a mucus. All of this you can see here is a mucus. And this mucus would drab around the chyme, would drab around the wastes, okay, and to make their surfaces smooth. So it progresses through the large intestine, it progresses through the anus smoothly, will not cause any damage to the area, to the tissue there. Okay, so we have plenty of goblet cells, and we have some absorptive cells that mainly will absorb water. Those are, we call them enterocytes. Those are enterocytes that they will absorb water, they will absorb salts, they will absorb the vitamins made by the intestinal bacteria. So all of this will happen in the large intestine. Then modification wise in the large intestine, it has 3 unique features. The first one is something called the tinea coli, and the tinea coli are 3 bands of smooth muscles. Okay, if you go back to here, see this line, this is the tinea coli. This is the tinea coli. So if you remember when we talked about the alimentary canal, we said we have circular muscles and we have longitudinal muscles. In the large intestine, we have the circular muscles wrapping around the entire alimentary canal. So anytime you cut, there are circular muscles, but the longitudinal muscles are only on one side of the large intestine. Okay, that is the tinea coli, and this will help in the progress in moving forward the wastes, because those will contract. So we kind of think of it like a sleeve. You are pulling the sleeve, and therefore the whatever there in the large intestine will move forward. So this is the first modification, the tinea coli. Then another modification, which is the appearance of the large intestine, those like pulps. Each one of them is called hastrium. Both together are called hastra. The plural form of it is hastra. Okay, what are those? Those are just in the appearance of the large intestine. Because of the contraction of smooth muscles, they will squeeze. So those lines are a result of squeezing of the circular muscles. This will help, in forming the feces and solidifying it or making it like a group of wastes. So that is the hastra. Then the third modification defecation, which is not shown here, is those the epibulic appendages. Those are appendages of fat, like, or sacs of fat, that they are around the large intestine, but there is no clear function for them. Why do we have them there? Almost nobody knows. So that is for the large intestine. Now, as we said earlier, the rectum will temporarily store the feces for defecation. When we are ready to defecate, it will process to the anal canal. The anal canal has two groups of sphincters or muscles. We have an inner smooth muscles, okay, inner layer of smooth muscles, and external layer of skeletal muscles. The inner muscles are involuntary, the external muscles or the skeletal muscles are voluntary. So when we are ready to defecate, the anal canal would help in pushing those the feces outside the body. So for the bacterial flora, we already talked about it. Those are bacteria inside our intestine. They will help in breaking down some of the undigested food, turn it into vitamins like biotin, vitamin K. All of those are important. There have been several researches related them to the immune responses, relate them to the ability to be obese or not obese, to diabetes, and so on. So all of those are in the bacterial flora. And of course, those are good bacteria as long as they stay in the large intestine. Outside the large intestine, they are damaging bacteria, and we'll deal with them as bacterial infection. Okay, so I'll stop here. In the following video, we'll talk about the liver and the pancreas.